patterns. And if you haven't already noticed, he's got tools set back up there where you can purchase some of his photographs, and there's a book that he collaborated on, and he will accept payment on any of that after his presentation. Some of you already bought a few things. He's an international miniature photographer and a naturalist based in Texas. He's out of Carrollton. And he has a master's degree in architecture from University of Texas at Arlington. And his specialty
Is this on already? Yeah. Okay.
Photography, exploring the flora and fauna around the world. There's some pictures of butterflies in that also. He loves to explore forests, parks, and gardens, and he's looking for both rare and common specimens to add to his collection. He volunteers with Fort Worth Botanic Gardens, and he is a Living Collections Associate, where he spends his time photographing and documenting the third largest begonia collection in the Americas. He's currently concentrating on
Am I gonna have to turn this one off?
And
You get her.
And he has recently put on an art exhibit at the Fort Worth Botanic Garden at Britt. And that's where some from our succulent team saw him and got him in touch with us. And so this is the hidden beauty of plant patterns and the geometric patterns, especially the Fibonacci sequence in nature. Any questions for him you can ask afterwards. And like I said, he has a table back there. He'll meet you back there after his presentation. So Victor.
Okay. Thank you for having me and giving me this opportunity to share my nature photography journey. I've been doing this for over 10 years, and doing the presentation, this is my third presentation. So yes, I bumped into a couple people from here when I was doing the exhibit of the hidden beauty of plant patterns in Fort Worth. And it was at the BRIT, which is the Botanical Research Institute of Texas. It was a display of a couple plants, more of the succulents and cacti, which those display more of the Fibonacci sequence patterns. And I will be showing you today more of the fauna and flora that I photographed. It's not just plants, but also do some animals, insects, macro, close up, and that's what I'll be talking about. But yes, so like, so you can see here, this is a cactus, and you can see some of these patterns. When I was looking at some of the cactus and succulents, I didn't really know what I was looking at. I was just seeing the patterns, and yes, they're going in a spirally way, but didn't really pay attention too much. After I collaborated on this book that was published by Princeton University Press, and it was this book, or it's the book that I have here in the back, Do Plants Know Math? And it's a collaboration of 4 co-authors. It's a writer, 2 scientists, and a mathematician. And this mathematician is one of the leaders for plant patterns. And so after collaborating with them, I learned so much from this book that I started seeing my images a little bit different, now understanding, and now this is what I want to share, for y'all to understand what I'm seeing as well. But let me tell you how I got started. So I started buying a few plants. And that was trouble. Wanted to put, I don't know, just a flower bed in the front lawn, and it got more intense than that. I saw the backyard and saw an opportunity to bring more plants in. And after bringing a few, I went back to the nursery and now I wanted this one, now I wanted this one. And here you see a garden in my backyard when I used to live in Fort Worth. Now I live in Carrollton, but 6 years ago, I was in Fort Worth, north of Fort Worth, by Keller area, Alliance Town Center area, where I lived there for 10 years. This is a garden that's been maybe 3-4 years in the works at that time that I took the picture. As you can see, some of the black-eyed Susans, the Texas rock rose, some cosmos, zinnias. You see a puppy there, that's my dog. He's no longer with us, but he used to just love running around the garden. Some marigolds over there. So this was, happening after I moved in maybe for 4 years, and then I moved out, and I pretty much transferred it to the new owner. I went to go visit my neighbor, and then sneak peek to see how the garden was doing, and it was doing good. The first year after that, I didn't do that anymore. But so this is still the garden in different perspectives. I got to now see that many visitors were coming. I started planting host plants and to attract many butterflies. And when you attract one insect that you think is gonna come, many other insects will come and follow. If you have a butterfly that is laying the egg and then the caterpillar comes out, well, some of the birds are gonna get some of those caterpillars as snacks, and spiders and so on. You can see these bunnies over here. My dog used to chase these bunnies. But they're cute, tiny little bunnies. You can see here the common buckeye, monarch, the Gulf fritillary, and then I got into collecting cactus and succulents. So here you see some of the cactus in bloom. Is an Echinopsis, the pink one that you see, and this one, the orange one, is an Echinopsis as well. But I just love the stem of the cactus, and also how the succulents, give you those patterns. And some people will collect some of these plants for the stem or for the flower. And you can see these flowers don't really bloom for more than 3 days. Some will stay on for a day or 4 hours. And I got just, crazy about collecting some of this cactus for their flowers. As you can see, so many colors, patterns, and again, going a little bit crazy on that. Oops. I don't know what happened. Okay. So this two in the middle is the same plant. And as you can see, the stem here, I don't know if I was trying to get the pointer. See, I don't know what's happening. Okay. Well, okay. I don't know what's happening, but okay. This right here, you can see that the flower is taller than the stem. You see the stem is a little bit smaller than the flower. And that's the same one over here. It's almost growing like a trumpet. And the one on top, this one over here is Trichoceras, is the same one. This one and this one, that's the same. Okay, this is what I was trying to get, a pointer. There you go, okay. So I'm using this camera, a DSLR, So after seeing so many insects coming into the garden, I wanted to see more of the smaller insects and other animals. So I couldn't see with my phone, and I would zoom in, and my quality was not as good. So I wanted also to record. So I invested on a DSLR. The first one, it was a D800, and with the macro lens, so I can zoom in a little bit more. And then eventually I upgraded to the D850 and another lens. So this image is one of the first ones that I took while I was walking at the botanical gardens in Fort Worth. And I took my camera, self-taught, didn't just watch a couple of videos. I took myself with the camera and the lens and started shooting. This was taken right next to the cactus greenhouse. And so they have a greenhouse, and they also have a cactus garden outside. And it's a beautiful cactus garden. It's been, upgraded. It's a little bit bigger now and more plants. But back then, I didn't know anything about plants or insects. So I was just shooting. Whatever was in front of me, I was, capturing that. So to me, when I capture this, in the camera, you have a viewfinder. The viewfinder is so small that you can't really see the details unless you zoom in. Well, I took this images to my computer at home, loaded those images, and I saw this on my screen. And when I saw this image, just seeing the bee and her face full of pollen, it just gave me another level of understanding the minute, small details in what's happening. Because if I was just to take a picture and see the bee and the flower, that's it, But after seeing more details and depth and zooming in, I was in awe. To me, I got hooked into macro lens and recording some of these insects and bugs. Well, I was focusing on the bee, but then at the same time, I was looking at the flower. The flower was amazing too, and I wanted to go back and now take a picture of the flower. Well, the flower was already done. The bloom was gone. And I went to the greenhouse, and I saw a sign saying that there is a cactus and succulent club, society. It was the Fort Worth Cactus and Succulent Society. So I joined the club, and I wanted to learn more about them. And they told me, yes, some of these flowers is only like 1 to 3 days. Some can be longer, but typically, general, it's about that length. So I was like, oh no, I have to wait a whole year or a whole season to get this flower, but it gave me a challenge. So after joining the club, I learned so much about so many plants. And again, you saw some of the collections that I was showing you. That comes from that, from being a part of the Cactus and Succulent Society. So I was also recording some of the insects that were coming, like the butterfly. This is the monarch butterfly. And I always ask people, and some of the people here know if this is a male or female. And people can tell by the 2 dots that you have on the hinge wings. And that's where the male monarch butterfly would distribute the pheromones to attract the female butterfly. But I have heard that on this monarch, the mechanism doesn't function anymore for the pheromones. So she still has the dots, but, he still has the dots. I call it she when it's a butterfly, but yes, I don't know too much about that. But okay, so I was recording. Me having the host plant is just waiting for the butterfly, the insect, to come and lay the egg. After the butterfly lays the egg, about 3 days later, the caterpillar comes out, starts eating part of the egg, and then the plant. I didn't take a picture of the egg here, but the rest I did when the caterpillar was on the milkweed. So it's a milkweed butterfly. And then you see it pupating. And it has a cremaster where it hangs. You know, it's almost like a, sorry, I don't know if my pointer is not working. But okay, so you see the stem where the caterpillar or the chrysalis is hanging from, and that's almost like a Velcro when the caterpillar builds this silky mesh around it to hang itself and pupate. It's very interesting just to know that that exists, and when you zoom in, you can see that, you know, cremaster is like a Velcro to grasp onto. And then you can see the last chrysalis is actually the same one, but it's from the front, and it's towards the last minutes of the cycle when the adult butterfly is going to come out. So the chrysalis is a clear shell, and what you're seeing now on the other one is just the colors. You can see some of the antennas and the wings, and you can see part of the butterfly patterns. But it's very interesting to see, you know, all these details and recording some of these butterflies. This is the black swallowtail that I also recorded. On this one, it was kind of tricky to understand the chrysalis. I always thought that it was going to change according to the background, but no, the color of the chrysalis is determined genetically, not by the individual in immediate surroundings. So this chrysalis is the same. It's just one taken from this side and the other one from the other side. And yes, you see it changing, but it's because it's aging. So this one is just aging, and yes, it starts this way, goes that. This is from the front. You see how well camouflaged the chrysalis is, and then over there as well. And then you have the adult butterfly. These are other insects, and like spiders and dense-fly, a moth from my garden, and this is a jumping spider. This is a dragonfly. Now, I started playing with the light. This is backlit. So this is a sleeping dragon, meaning it's not the name of the dragon, but it was during the night. And what I did is I got 2 flashlights from the back, and I wanted to see and show you the patterns on the wings. And you can see kind of detail of the wings, and started playing with different positioning, you know, lighting in different ways. So for the still life macro setup, and when I say still life is for plants. Most of the time that I shoot some of these cactus to show you the patterns, it's a still life macro setup. And this is what I have been using. It's a light box. And then you have a tripod. And then just to reach over with the tripod over the box, I use this adapter, horizontal adapter, and then trigger. So this is some of the setups that I have done, also including this attachment lens, just to zoom in a little bit closer. And this is a setup that I have at home and also at Fort Worth Botanic Gardens at the Brit, where we are recording some of the begonias. And you can see here in the middle that I am photographing this small pink flower, and that's how close we get. But the images, after you focus stack, this is for focus stack purposes. It's amazing. I mean, you zoom this image, and it's the size of the canvas here, and it still holds that detail. So this is some of the software that I've been using: Serene Stacker, Helicon Focus, and Photoshop. Photoshop doesn't really work for me, but the first two have done a really good job on focus stacking. So when I talk about focus stacking is a method of photographing some subjects. And for example, if you ever look through a microscope, you have a subject and you're looking down to the subject. Let's say the subject is this big. Well, you can't really see the entire subject when you're shooting with a microscope. You only see a very tiny portion of it. Is the depth of field that you see. So when you photograph this images or these subjects, what you do, you have to take multiple images. For me, for this cactus, about this big, 2 inches by 2 inches, and you take about 30 images. You combine them in a software, and then you create the final image, where the final image will be crispy, clear and very detailed from top to bottom. And that is the purpose, to do focus stacking, to just bring more of the detail and show more of the patterns. So this is an example. And this one, it's a mammillaria scopa. It's a cactus. This is about 30-something images for each. So this is from the top, this is from the side. And as you can see, all the spines will be in detail. If you were to take just one image, part of the cactus will be in focus and then the rest will be blurred. So with this, you can actually print it the size of the wall and have it, in detail. When I was zooming in for the first time into these cactus with the microscope or with the macro lens, I was seeing multiple insects, very tiny insects. And I got to see up to 7 or 8 different species of insects. So when I was looking in and zoom out, to me, it gave me a different perspective. It was almost like their own world. And these inhabitants were living there. It gave me a different perspective. But also the beauty of seeing just the pattern is more present. And you can see on the other one as well. This is another example of how detail is from top to bottom. You can see more of the patterns. And this is how I will take it inside the box. This is the mammillaria laui, and it's about 2 inches by 3 inches. And when I take it in, I just improvise with pieces of wood blocks and paper, and then center it to the camera, and then start shooting multiple images until I finish, from top to bottom. So again, these are the results that you get, and then you can zoom in, and you still have that sharpness. Those spines will be in focus, and you can even see at the bottom the buds, the flower buds here, those light green buds. So another method that I use now when I wanted to take it outside of my garden or outside of the studio, I wanted to have an image that was not harsh with the flashlight or with the natural light. In some cases, a diffuse light, it's much better quality to eliminate, just the harsh highlights and deep shadows, like the one in the middle. And this diffuser helps to take your camera handheld and take it outside of the studio. And you can get very close to the subject and will be kind of lit up evenly. So this is, the example, diffuse light, the same beetle. And so for animals that are reflective, for eyes, for like the beetle that you saw that is a little bit too reflective, if you don't have a diffuse light, then you won't see the details on some of these reflective subjects. So this is an example of a frog, and then you can see that the eye doesn't have those harsh highlights. And it helps to show you more of the details. This is a cuckoo wasp. Now this cuckoo wasp, was living in one of the mud dabber nest. And so this is a parasitic wasp. It's a solitary wasp that doesn't really build their own nest. So they go and use other abandoned nests. So she was there inside the garage. I wanted to knock that mud dabber nest down, and she came, down. I didn't really see what it was. I had the garage open. The sun was hitting this metallic, subject, and then I realized that it was a wasp. It was a beautiful wasp that I'd never seen, or never really paid attention. And so it was dead, and it was curled this way, and I wanted to record it. So then when you do that diffuse light, again, you can see more of the detail, and just better representation when it's a metallic, iridescent subject. So you can see these details. Now, I wanted to take this outside of my garden, outside of the parks that I go visit, and typical gardens that I used to go visit. It was the Dallas Arboretum, the Fort Worth Botanic Gardens, Lila, it's over there by the Louisville Lake, and many other, gardens here that are local and preserves. But now I wanted to go and explore other places, other countries, and see other insects and animals. So I went to Costa Rica, and this is the red-eyed tree frog, which is very iconic to Costa Rica. And she was making a call, or he was making a call, and I saw from behind, I was in the back, and I saw the frog came towards the front and snapped that picture. And again, they're gonna be wet most of the time. They're very reflective, but if you have that diffuse light, you don't see those harsh highlights, which makes a nice image. Now, again, you have more different species from Costa Rica, and you see these two frogs on the side. Those are poison dart frogs. They're very tiny, and one thing that I learned about the poison dart frogs is that those are diurnal. They come out during the day, and the other frogs come out during the nighttime. So if you were to look for them at night, they're hiding, unless they're mating and they're making a call, and that's how you can hear them and find them. But the one on the right-hand side, it's a very endangered species that I learned another thing, taking pictures of these small insects in search for animals that are in danger. So when you go and visit their place, you have to be aware of your ethics as well. Well, what is it that you're have to be aware of? Some of these frogs are actually dying from this fungus. And so if you're stepping into their habitat, coming from another habitat, you can transfer on your boots some of these fungus, and then you can harm the ecosystem. So you have to spray your boots, disinfect your boots, and just make sure that you don't bring anything from outside. And that's part of the ethics as well, not harming them, not touching them. If you were to, because you're doing a study or something like that, then you wear gloves. You don't want to touch them with your hands. You don't want them to harm you, and you don't want to harm them. Well, the oils on your hands can harm them as well. And then they're poison dart frogs, but they don't really have the poison on their body. If they feel harm, they will excrete their toxin, and then that's for protection. But yes, a lot of people think that just by seeing them, they're gonna get, harmed. But no, these are creatures that are, very beautiful creatures that are, hard to find, and also people don't know, they're not aware. Another thing that I learned by taking these images and showing people is making them aware of their existence and their habitat. We're seeing that many of these areas are being wiped out. And so if you don't bring awareness, if you don't record what's in there, anybody can come in and just wipe that area. So that's what's happening in Costa Rica. Costa Rica is one of the leaders in preserving some of these habitats. And it's also part of the tourism that is helping the country. For example, if you want to go visit that frog, it's an area, or it's several areas that are protected. To go visit, you have to have a guide to go in, and then if it's like an Indian reserve, that guide has to be with you. So it'll be sometimes two guides watching out for you and the species that you're visiting. And it just creates an income for them, generating, tourism. And to me, it's a win-win situation. We're protecting them. I believe there was 5 to 8 different species of these guys, and only 3 exist. The other ones have been wiped out because of deforestation or some of the fungus being infected by. This one I love because this green and black frog, dart frog, was on an orange leaf that loved the contrast, just the different colors in it. This glass frog. Again, it's more from Costa Rica. This katydid looks like a leaf. And when you see it, first you think it's a leaf, but it's a katydid. And this frog over here is the blue jeans. That's because of the color of the legs. You see the blue, and the common name is the blue jeans. And this is a eyelash viper. This is the eye of the red-eyed tree frog. It's just kind of closing this frog. Now, you see on the right-hand side, amplexus is when they're mating. You have the male smaller on top, and the female a little bit bigger. And this frog, there's 2 different, morphos. One in the Pacific, and then the Caribbean. There are 2 different morphos, meaning their patterns and colors change a little bit, and that's how you can distinguish. Like this one is from the Caribbean side. It has the blue on the side, the blue colors. And then the one in the Pacific has more yellowish, no blue. Now Peru, again, some other frogs and just beautiful grasshoppers, very colorful. And this frog over here that you're seeing, the Calimedusa. Okay, it'll be on the next slide. But again, you see the eye there with no reflection. Use a diffuser to get close up and see the details on the eye. The blue morpho caterpillar. You can see this frog with the sac making the call, which the males are the ones that make the call. So you know that this is a male. The frog that I was telling you, this Calimedusa, I was recording with this university in Iquitos, Peru, where this group of people were recording 250 plus frog calls. And they were wanting to do that to put the call into a picture and people will know if they hear that's that frog. So they were doing that. While they were doing that, they were doing other recordings, other investigation, and I collaborated a little bit on that, where I wanted to record this frog, the underneath side, how they, put themselves on the leaf. Here's camouflage, you see it from the side, but then I wanted to see how they look when they're hugging that leaf. Well, the way we did this is with the glass, and then she'll, sleep or be calm, and then that's how you can, see that. And then she's moving around. That's not a jump, that's just walking around the glass, and we, snap that picture, and then you can see the patterns that you can't really see. This is Brazil. Brazil bumped into a lot of the beetles, and yes, there's a lot of spiders. I know a lot of people don't like spiders, but there's beautiful, beautiful spiders out there, many moths. And when you see black background versus this natural environment and natural light, you can see that the ones in black background is shot most of the time during the nighttime with the diffuser, and these are during the day. So you can see that here on top on the left-hand corner, this dragonfly is very iridescent blue. And it's just like amazing to see it in person, because when this dragonfly closes its wings, on the other side is almost black. There's no color. But when the wings are open, it's just this iridescent blue color. And yes, it has to do a lot with the structure of the wing and how some of the light bounce back to our eyes. But it also depends if it's forecast or if the type of natural lighting that is happening, or the tilt on the wing, you might see different colors. So you have to kind of be patient to see the most iridescent blue and kind of capture it, because one angle you might not see it, then you go to another angle, you might be able to see it. So I was playing with that, where you can see the just true iridescent blue color. And then you have some leaf hoppers, this strange looking tree hopper. It's just, to me, a very rare insect. Now this one in Mexico, I don't know if you can see the pattern on this butterfly, it's the 88, and that's the common name of the butterfly, the 88. And so there's times that this butterfly would develop a 9 in the front or in the back instead of an 8. So sometimes it'd be 98 or 89, but the common name is 88. Most of the time she's 88. And I tell people, oh yeah, there's 1 to 100 numbers on some butterflies. And they believe me until I say, no, it's not true. But there's some butterflies with some numbers, and I'll show you some here in a minute. This glasswing butterflies, they're, to me, stunning, because I also learned that these glasswing butterflies are not reflective. The structure, again, on the wings makes it non-reflective. If they were to be reflective, it would be easy for birds or other, predators to come and get them. And so these butterflies live under dense forest. So there's not a lot of light. That's how they camouflage themselves. They're almost transparent. And the only thing that you're seeing, because it's dark, is that white line, that stripe. You see that other one is the different one, species, a different species. You see the other one has dots, this one has that stripe. But yes, that's what you're seeing, the little stripe going, and that's how you find them. But they're hard to find. Well, then how do I photograph these two? Well, they're resting during the day. They don't really like harsh sun, but they have to eat. They either come out during the morning when the sun is not, brutal, and at nighttime, that's when they come out too and start eating some of the nectar from the flowers, and that's how I was able to photograph them. Again, some more from Mexico. This one over here in the middle is the Mexican blue wing butterfly, and that's one of those that, depending on the angle, that's the colors that you will see. Sometimes you'll see blue, sometimes you'll see purples. Here's like a mix of blue, purple. And then here, Texas, Lila, I've visited so many times. There's other gardens that I have visited. The caterpillar in the center, that's at the Fort Worth Botanic Garden, one of the trial gardens. And again, more from these parks. This one is 2 dragonflies. One is eating another. I don't know if you can see that. This over here is a longhorn beetle that was on one of my apple trees. And then again, more. You see the caterpillar over there. That's one of those that you don't want to touch because some of the spines are very toxic. But yes. Again, now kind of transitioning from fauna to flora and the mix of patterns. I was seeing on ammonites these spirally patterns, and then on the petals. And then I don't know if you can see on this over here image at the bottom, there's a pattern. And I tell people that sometimes people say that those are penguins. Let me see, I don't know if I can, well, I can't zoom in, but I don't know if I can see this over here. These little ones over here, they almost look like penguins, little penguins. Yeah, I don't know what's happening. I was able to zoom in, not anymore. Oh, okay. So, yeah. Now, if you look in the top right-hand corner, you see another butterfly with numbers, and that's the 80, and I've seen the 86. And then somebody asked me, is that a 1,111? I was like, no, it's just stripes, stripes. Now these over here also have these stripes, and as you can see, some of these will be branching patterns, spirals, cracks, spots, stripes, so many symmetries. And again, with symmetries, some mushrooms and some of these sea pods. And then, again, some of these patterns or colors is to camouflage themselves, like the eyes on some of the moths. People say that represent like owl eyes. This one is almost like same color as the leaf. I don't know if you can see this grasshopper, and he has the antennae. And this over here, those are eggs. And those eggs are, I believe, from a kissing bug, if I'm not mistaken. And what I learned about these eggs is that if the kissing bug puts the eggs underneath the leaf, the eggs will be white. And if they locate them on top of the leaf, they'll inject this dye, this dark dye, to protect from UV light and not burn the eggs inside. At the same time, these little spikes is maybe for protection from predators. And then you can see the camouflage frog over there on top of the leaves. Now, I don't know if y'all can see the camouflage insects, but the two on the sides, those are mantis. And then the one in the middle, I still don't know if it's a cricket, a katydid, or a grasshopper, but I think it's not a grasshopper. It's one of those katydid or cricket. Now, again, recording plants. I was recording this in Costa Rica, and that's a fern, and it got my attention, and that's the one that was actually used for the cover of the book, Do Plants Know Math, where I have some of the books over there in the back. But this is the book that I was telling you that I collaborated with these co-authors. I'm just the main photographer, so many of the images were used for this book to explain some of the patterns. And I guess they chose my images because the way I was just showing the patterns and the detail. So this book explains more on the spiral side of the patterns, and it goes, tells you the history all the way from Leonardo da Vinci when he was paying attention on some of the plant patterns that was happening. So, I don't know how I'm doing in time. Okay. To understand some of the spirals, there's about 28 different spirals, and some of these spirals have to be explained. A mathematician has to describe them so you would know what a spiral is. And all these spirals are a different type of spiral, but then at the bottom you can see there's the Fibonacci spiral and then the golden spiral. So you can see some of these spirals and some shells, some plants, some of the horns, the double helix in our DNA, some of the plant patterns again. And you have these two that I'm going to be talking about. You have the Archimedean spiral, here the first one, and then the logarithmic spiral. Now the differences between this is that on the Archidemian spiral, it's a constant, the thickness of the, for example, the rope here, it doesn't change. The width is the same width, it doesn't change. So it's constant, spiral on the width. This one says the same thing, the millipede, the millipede. But just because the legs are covering the body, it looks like, logarithmic, but it's actually the same width. The body of the bug is the same. And then the logarithmic, it is changing the width, but it's the same. It's just getting bigger and bigger. So that's a logarithmic spiral. And again, this is the Fibonacci. Now, the Fibonacci spiral and the Fibonacci sequence is different. When we talk about Fibonacci, a lot of people get confused with Fibonacci sequence, Fibonacci spiral, Fibonacci. There's so many, variables that you can put, but the sequence goes like this. It's pretty much the series of numbers where each number is the sum of the two preceding ones. So if you do 1 plus 1 is 2, 1 plus 2, 3, 2 plus 3, 5, and so on. That's how you get the sequence. Now, to jot down the spiral, the Fibonacci spiral, you see in the middle with one square. So 1 plus 1, you see the 1, and then another one is 2. And then on top is 2, so 1 and 2, 3. Then you see 3 on the side. And then 2 and 3, 5, and so on. And then you throw that arc diagonally and keep going, and that's the Fibonacci spiral. So if you go online and search Fibonacci spiral, you can debunk a lot of these concepts where people are trying to tag so many things with Fibonacci, but this is the true Fibonacci spiral. And the sequence is different. It's the sequence of numbers. Okay. So you can find these sequences in many plants, in botany mainly, not in animals. It's very rare, or maybe none. But in plants, you can find it in pineapples. You see the spirals on the pineapples, artichoke, pine cones, and then also on the petal count. So I went to Barnes & Noble and grabbed one of the books for Texas, or I think North American wildflowers. And I was just flipping through the book. And yes, a lot of these flowers or plants show on their flowers a petal count of more on the 3, 5, 8. But again, those numbers are part of the Fibonacci sequence. And again, not all plants or flowers have this count, but they're saying that almost 90+% of plants have it either on the petal count, on the bud, or seeds, or the way the plant grows, the leaf distribution, and so on. So again, phyllotaxis is, botanists will call this the arrangement of leaves and flowers in plants. So for example, in this succulent, the distribution of leaves, you can call that phyllotaxis. And again, to just understand and go more in depth, the plant's tiny organ factory is called the shoot apical meristem. You see it over there on the right. And that's where the base where the plant growth is happening. And then the primordium, it's one of the bumps. It could be a petal, scale, or a tubercle on a cactus that is growing. And if you count all of them, the plural is primordia. So another concept or vocabulary will be the divergence angle, and this pretty much explains where you have the first leaf and where's the other leaf gonna grow, and it grows at 137.5 degrees. You see it 137.5 degrees, that's the golden angle. And so that's how the leaves are gonna be distributed on this succulent. And then you have another level of understanding spirals. If you count the spirals going one way, counting the primordium, you find one spiral, this one 2 spirals, the other one 3 spirals. And those spirals in botany scientists call them peristiches. So one of these spirals is called peristiches, or peristiche. And when you count the spirals on, for example, this pine cone, it's the same pine cone, and you see the bumps, right, or the scales, primordium. If you count them clockwise, you get a count number. This one is 13. If you go counterclockwise, then you get a count of the neighbor number of the Fibonacci sequence. So it will be an 8. So this is a count of 8. And it goes, again, clockwise, counterclockwise, and so on. It goes both directions. Again, this is just to show you that the anatomy of the cactus, tubercle is these little bumps that come out, and that's the primordium. And this is an example of just one of the images that I've done. This is about 25 images combined to create the last one. And then you can see more of the patterns. You can see the spirals now. Again, count on some flowers, pineapples. You get count of 34 here. If you count them the opposite way, counterclockwise, 55. Pineapple, again, just to see what the numbers are with the string. I counted one way, you count 8 spirals. Count them the other way, it's 13 spirals. And then you have the crown, on top, just doing the same thing. Each of the dots or the scales on the pineapple, those are called eyes. And each of the eyes are fruits. So it's almost like a fruit that has a lot of fruits. And this is fractals. This is Romanesco broccoli. It's a good example of just showing how some of these heads keep repeating themselves. And here we have it from the top and showing you how some of these heads are almost the same as the next one, and this keeps repeating itself. So as you see, the smaller heads, it's almost like a replica and a copy of the same. So one of the scientists got to count 7 different heads going down and getting that same pattern. At the same time, you have the Fibonacci count on the spirals. And so one is 8 and the other one is 13. Now, if you cut a red cabbage, you also see the spirals. And this cabbage is actually an enormous bud that hasn't been opened. How do you know that? This is just to show you different colors, patterns, symmetries. It's not just, spirals, but it's also shapes. So here you have the oxalis that has the triangles. This astrophytum, you can see some of the squares, and so on. The stapelia has the 5 pentagon, and the 8 count polygon, the octagon, the gymnocalycium, and the parodia. They're just different shapes on some of these. Again, now this helps you to distinguish these peristiches, the spirals, visually, because it's separating the bumps or the tubercle, with the woolly hairs, and it's almost helping you to count the spirals. If you were to count the spirals now, I have done that here, where I can get a little bit extreme. Well, if you start from the bottom, and it's a little bit complicated at first, but you can see that I found a count of 3, 5, 8, 13, 21, and 34, and that's all Fibonacci sequence, and is naturally growing that way. Okay, how do I find them? You see this, I went with the strongest visual, the one that you can see, which is 13, and then I count the opposite side now, counterclockwise, and find 21. So when you merge these two to find the next one, the intersection where they cross, that's how you find the next one. And that's, again, Fibonacci number 34. 13, 21, 34. And been doing it for the rest of the plants, and I always get these numbers. Now, not always. There's also the double Fibonacci. So double Fibonacci is just double the numbers, and that's what you get. And then there's the opposite, where it's going to be one of those three. But most of the time is the Fibonacci count. Again, with the succulents, with the cactus. Now, this one I'm showing you just this way. I wanted to represent it, and it was inspired during the eclipse. And so I wanted to backlit this cactus. It is a mammillaria. So you can see the honey color of the spines, and you see a variation of these spines. Some are hooked, some are straight. And I just bounced the light so it can be a little bit dark in the middle. But yes, I've done this Fibonacci count, and it has it too. Now, I've also been doing a project here recently with the Fort Worth Botanic Gardens, the Begonia project, with the Begonia Society in Fort Worth. And we're recording one of the largest begonia collections in the world, where we're recording all the species and some of the hybrids. These will be, going through their season, and we're recording the male flower, the female. We section the plant and do the seeds, record the seeds with the with the SEM, which is the scanning electron microscope, and see the tiny as like the pollen, and it's just beautiful to record that. And the reason why we're doing that is because there's not really a complete database for the begonia plants online or anywhere. And if there is a database, it's incomplete or poor quality of images. And so we're trying to have this collection to help future scientists or people that want to do research. And so far is me leading this project for the photography side, and then I'll be having interns or volunteers that want to come and help, and we do it throughout the season and record some of these plants. So this is what we do. We record just different types of species and different ways of recording. This is some of the plates that we do as well. So we record like the female flower, the male flower, break it down, have dissect the stem or the ovary, and then the seeds, part of the stem as well, and the whole plant. This plant right there was about 6 feet tall. And yeah, you can't really tell the scale. I need to put scale on these, but it's a little bit complicated when you have different zooms and trying to get everything on the plate. but this is what pretty much we are doing for each of the plants. So this is a long project, it's a long project. And that's pretty much it. Thank you. All right, so I'm open for questions and or any comments. Yes?
When you're in Brazil or wherever, how long do you spend out there?
So most of the time, I'll try to stay longer than 2 weeks to up to a month. It's kind of ideal to spend a good time. So for example, I went to Peru and I went to the reserve of Manu. Manu, this is the trip. From here, Dallas, you get to Cusco, or sorry, Lima. And then from Lima, you get to Cusco. You get to acclimate yourself because you're in high altitude and you can't really walk, because you'll get tired. The oxygen is different because of the altitude. So you have to acclimate yourself. Then from there, I went the next day up the Andes, down to the Amazon area, where we had to drive 9 hours to get to the Amazon. And then on a boat, the next day, 9 hours. The next day, 9 hours on a boat again. And then the next day, 2.5 hours to get deep, deep in the Amazon. So that's just 5 days going, 5 days coming back. That's 10 days. So that I needed a month to get stationed deep in the Amazon. So that's just one of those extreme examples. But other places, for example, Costa Rica, 4 or 5 hours, get there. But then, let's say I want to see some frogs, right? Okay, how many frogs do you have? 156. How many poisonous dart frogs? 60 or 40. Okay, I want to see how many I can see, right? But just to give you a perspective, they'll tell me, okay, here in DFW, you have 6 frogs. Then you have to travel to Houston for another 2 frogs. And then, another area, Houston, for another 5 frogs. So they're not on the same location, so you have to move around. At the same time, go with local guides that are experts in finding them, either by call or knowing, when is the right moment. When the moon is out, they come out. When it's raining, at nighttime, and where there's frogs, there's snakes, so be careful. But the guides know how to, get there and help you what not to touch, like some plants and so on. So it is, not easy, but at the same time, if you put dedication and time, you'll be able to find some of these cool insects or frogs that, you would never really plan to find. Yes?
You're taking a picture of like that jumping spider.
Yes.
I know you're zoomed in, but
Yeah.
how close are you and how do you get him to sit there?
Yes. So there's a couple methods. There's you can do stacking handheld with a monopod. So it's just the one stick, right? And you're actually kind of measuring the depth of field, and you're moving through the insect. So one shot is here, the other one's here, and so on. But it's not easy because when you're that small, and any movement, like you're breathing, plays it moves you, and then the trigger moves you too. Your stand moves you. So you have to be very steady, and you can do it by doing handheld stacking. But the one that you saw is one of those special occasions because it was a pet. And that one, it was in my garden, and I wanted to find out more about jumping spiders. So what I did, I learned that they go through some molting, like 5 stages of molting. They had to be in the right space for them to be healthy and also to hunt like they normally do outside. So I'll bring crickets, I'll buy crickets from the pet store so she can hunt them, or flies. They love flies. And they're triggered by the vibrations. Some of the hairs, help them tell that, oh yes, that's a meal. Because I tried to give her a dead fly and she would not go for it. Only for the live ones, the ones that are flapping. Yeah, so but I learned so many things about it. But yes, that was one of those that I did in the studio, that one. Mm-hmm.
Have you tried in-camera stacking as opposed to post-processing?
I have not, and I think I have a little bit more room to play with when I do it. Post-processing, because the way I'm doing it is I do it with the screen live, and I'm checking everything versus, oh, I should have done this and it's gone. I can't do anything about it. Yes. Anything else?
Amazing.
Thank you. Thank you.